Now the today's topic is HMP pathway. It is also called as Hexose monophosphate pathway or Pentose phosphate pathway. HMP pathway or Hexose monophosphate pathway or Pentose phosphate pathway. This pathway is also called as Hexose monophosphate shunt pathway or also called as HMP shunt, SHUNT. The term HMP pathway is due to the involvement of hexose sugar, 6 carbon sugar, glucose. So it is called hexose monophosphate pathway, and the name pentose phosphate pathway is due to the formation of pentose sugar, ribose from the same pathway. So one may call this pathway either HMP shunt, HMP pathway or pentose phosphate pathway. Both the names for the same pathway. Hexose monophosphate pathway depending upon the involvement of the 6 carbon sugar glucose, and pentose phosphate pathway due to formation of the 5 carbon sugar ribose. So this is about the why the name is given HMP pathway or HMP shunt or pentose phosphate pathway. So this, as the name suggests, hexose sugar is involved in this metabolic pathway. The name of the sugar is glucose. Glucose take part in HMP pathway in the form of glucose 6 phosphate. So there is involvement of the glucose 6 phosphate in the reaction, and there is formation of ribose 5 phosphate as a pentose sugar, which is involved in the nucleic acid biosynthesis, or which is required in rapidly dividing cell or rapidly growing cell where mitosis is going on and new nucleic acid synthesis is required. In that cell, this riboses take part as a building block molecule for the formation of DNA, as well as it is required for RNA, because DNA contains deoxyribose, RNA contains ribose sugar. So the importance of HMP pathway is to make available ribose pentose sugar for the synthesis of nucleic acid as a part of DNA or as a part of RNA. Now, during this metabolic pathway, though it is meant for the formation of the ribose pentose sugar, along with this, there is formation of reducing power NADPH plus H plus. This NADPH plus H plus, this reducing power, it is required for reductive biosynthesis of lipid. So for lipid synthesis or fatty acid synthesis, NADPH plus H plus molecules are required, and they are generated from this pathway. So NADPH plus H plus is required for synthesis of lipid. They are also made available from this HMP pathway. So we are going to study how this pathway is operated and how it is made available ribose sugar for nucleic acid biosynthesis. What are the different modes of the HMP pathway depending upon the demand of the cell, depending upon the need of the cell. There are different modes of the HMP pathway, and the cell supply different component as a NADPH plus H plus for lipid biosynthesis and ribose for nucleic acid biosynthesis. So some different situations, 4 different situations may occur inside the cell. In one condition, cell require both NADPH and ribose. In one condition, cell may require only NADPH, not ribose. In one condition, cell require only ribose, and in fourth condition, cell may require ribose, NADPH as well as energy charge. So how this pathway is operated depending upon the metabolic situation in the cell. So there are different modes of this HMP pathway. Before going to one by one different modes of the HMP pathway, we are going to study the 2 phases of this HMP pathway. The HMP pathway is divided into 2 phases. One phase is called oxidative phase of HMP pathway, and second phase is called non-oxidative phase of HMP pathway. So HMP pathway is divided into two parts: oxidative phase of HMP pathway and non-oxidative phase of HMP pathway. The first one operated is oxidative, second one followed is non-oxidative. Before going to the actual different phases of this HMP pathway, we are going to study the general reactions of this HMP pathway. In general reaction, as there is involvement of the glucose take part in the reaction in the form of glucose 6 phosphate. So the reaction is glucose 6 phosphate is converted to 6 phosphoglucanolactone by the enzyme glucose 6 phosphate dehydrogenase. There is removal of H from the structure of glucose and the removed H accepted by NADP to form NADPH plus H plus. I again repeat the reaction. Before going to the intermediates of the reaction, just vaguely we draw the general equation like this: glucose 6 phosphate, one molecule when converted to ribulose 5 phosphate pentose sugar, there is formation of two molecules of NADPH plus H plus. So as one can talk with respect to the importance of the HMP pathway, NADPH plus H plus molecule 2 are available when one glucose is converted into one ribulose sugar. So glucose is the 6 carbon structure sugar converted to 5 carbon ribulose 5 phosphate. So there is removal of carbon in the form of CO2, and at a time during this reaction two molecules of NADPH are produced. I again repeat the same reaction. Glucose 6 phosphate converted to ribulose 5 phosphate with formation of two molecules of NADPH plus H plus. So when one single glucose 6 carbon structure converted to one 5 carbon ribulose sugar, there is formation of two molecules of NADPH plus H plus. If in the cell they require NADPH plus H plus for lipid biosynthesis, then NADPH is available by this reaction. Ribulose is isomer of ribose sugar is also available because it is required for nucleic acid biosynthesis. The first committed enzyme in the conversion is glucose 6 phosphate dehydrogenase. So this is the committed enzyme in the reaction which convert glucose 6 phosphate to ribulose 5 phosphate. So this is about the outline of the general reaction, how the 6 carbon hexose sugar is converted to 5 carbon pentose sugar. I again repeat the hexose monophosphate pathway name on the basis of involvement of the hexose sugar glucose 6 phosphate, and pentose phosphate pathway is also called to the same pathway because there is formation of 5 carbon ribulose sugar which is nothing but pentose sugar, so the name is pentose phosphate pathway. This hexose monophosphate pathway or pentose phosphate pathway divided into two phases. One phase is called oxidative phase of PP pathway and another phase is called non-oxidative phase of PP pathway. In non-oxidative phase, there are interconversion of sugars takes place, which we can see in next. And in oxidative phase, These are oxidation type of reaction takes place. As we mentioned in the first reaction, glucose is converted to ribulose, pentose sugar, and during this oxidation reaction, whatever H removed from this glucose phosphate, they are accepted by NADP to form reducing power NADPH plus H plus. All students take a note that whatever the NADPH plus H plus they are generated, these NADPH plus H plus are meant for reductive biosynthesis of lipid. They are not like that of NAD, so they are not giving their electrons and protons to the components of electron transport chain. They are required for lipid biosynthesis. In lipid synthesis, we are studying in detail how NADPH is available for the growing fatty acid chain. So I conclude this general explanation as HMP pathway is hexose monophosphate shunt or HMP pathway, or it is also called as pentose phosphate pathway. The significance of the pathway is provide the ribose sugar for nucleic acid biosynthesis. It provides NADPH plus H plus for reductive biosynthesis of lipid. So this is about in general introduction about how HMP pathway is regulated, what are the different oxidative non-oxidative phases of the pathway, and glucose 6 phosphate involved as a first substrate. By the glucose 6 phosphate dehydrogenase, it is converted into ribulose sugar.